Hello and good afternoon friends, welcome once again to the CEC-UGC live lecture. Dear friends, this is the third and the last lecture on cell respiration, and under this lecture, that is under this series, today we are going to discuss about electron transport and ATP synthesis, and for this we have again with us in our studios Dr. Eklavya Chauhan. He is an associate professor in Department of Botany, Deshbandhu College, University of Delhi. So, taking advantage from his experience, as well as I would like to tell you all the way he has delivered his previous lecture with ample of examples, which made students to grab all the content which he has provided in the previous sessions. I hope that today also he is going to make this session very interesting, very lively. So, first of all, I would like to welcome our guest, Dr. Eklavya Chauhan. Dr. Chauhan, welcome to the EduSit lecture.
Thank you very much, Deepika. I'm delighted to be here to explain some of the most intriguing aspects of respiration, which are entailed in part 3. And this is a very complicated mechanism. Let's go ahead with it. If you remember, viewers, in the past 2 lectures that we had together, I had dealt in details about the glycolysis and the processes of Krebs cycle. If we were to recapitulate what we had seen earlier, we had a detailed account of glycolysis, and we found that only 2 ATP were gained in glycolysis, and there were 2 NADH which were produced. In the whole process of Krebs cycle, there were only 2 molecules of ATP which were produced directly, and again a large number of NADH and FADH2 molecules were produced. And I have been telling you repeatedly that we would be dealing in the last part of electron transport chain. How does this indirect release of ATP come into being? We must appreciate the fact that out of a total of 36 or 38 ATP produced, it's only 4 ATP that have been generated directly. So, today's lecture is going to be devoted on the theme as to how do the remaining 32 or 34 molecules of ATP being generated indirectly, and hence the topic being the electron transport chain and oxidative phosphorylation. Before starting this whole episode, let me just make you familiar with some of the very frequently asked questions by students as to what happens to the NADH and FADH2 which are produced during glycolysis and Krebs cycle. Because they are produced, they have to go into the mitochondria, and what are they going to do in the mitochondria, and how they are going to generate ATP. The second question, which is very pertinent, is that in the first place, what is the rationale of having the electron transport chain in the first place? Why can't simply the electrons and protons combine together? Why can't they directly combine together and give rise to water? The answer to this simple question is not that simple. We will look at it in details, and then this also would stem out another answer as to what is the need for the electron transport chain. Then we are always torn between the age old question of 36 and 38 ATP. We have to make a threadbare analysis of what does this mean. Is it always that the net gain of ATP in organisms by the oxidation of one molecule of glucose is 36? We know that in many cases it is even 38. So, what are the conditions which prompt the production of different types of productivities, and the process of ATP generation itself? How is it that a form of electron just passing through a series in a membrane can bring about so much of energy change which would be sufficient to couple a ADP molecule with inorganic phosphate to generate ATP. So, that means what kind of a process is this? Is it simply a chemical reaction as it seems at its face value, or perhaps there is some involvement of the osmotic gradient? Perhaps at the end we are going to reach a conclusion that it is something of both. So, we would like to call this as chemiosmotic coupling. So, that is the basic idea. Just to summarize, what we found in the earlier classes was that okay, ATP is formed directly and some of the molecules are indirect, and when they go inside the mitochondria, then they would produce ATP. Two things need to be known. Number one, what is the location of this entire process that we are talking about? The electron transport chain and oxidative phosphorylation in eukaryotes is going to be carried out in the inner mitochondrial membrane, whereas in prokaryotes there is no internal compartmentalization. So, we would find that the whole process is also being carried out, but then it is carried out on the plasma membrane itself. If we look at the process of oxidative phosphorylation, it involves two essential processes. Number one, the electron flow, which has to be according to a system, and phosphorylation, which means ATP synthesis from the precursors or the ribonucleotides ADP and the inorganic phosphate. One should remember that both these processes are absolutely coupled. They cannot take place in place of the other, although we have some exceptions. We will like to talk about it in the middle of the discussion. Then basically the problem of oxidative phosphorylation is oxidation reduction. So, first we have to understand what is the whole idea of oxidation reduction. The NADH which has been formed has to lose electrons and then it will be converted into NAD+. This is a case of oxidation. Simultaneously or a little later, oxygen has to be converted into water, and this can be done or this can be achieved only by the gain of electrons. So, that means it is reduction. In the final analysis, we would find that it is a redox reaction where NADH gets converted into NAD plus and half a molecule of oxygen with electrons gets converted into water. So, the summary wise we would say that the stepwise removal of electrons from the substrates which ultimately reduce oxygen to water, once this process takes place, then the protons are imported back into the mitochondrial matrix and the formation of ATP takes place. If one has gone a bit too quickly over the concept, one can even summarize this whole concept in a simplistic manner, and that would be energy which is released by the movement of electrons is finally utilized for proton pumping across the inner membrane and of the mitochondria, and this is how ATP is formed. Let us first look at the transport of electrons and protons. How do they really move? And this casts doubt on the term electron transport chain itself. Many workers have cast doubt. Why do we call this as an electron transport chain at all? Because our idea of transport is that it should take place across a membrane, but we find that the electrons actually move through the membrane. They are going to pass from one electron carrier to the other in a series of steps. We will just discuss why a series of steps, why they can't simply jump one step and be through with it. So, they have to necessarily pass through an electron transport chain, whereas the protons, they will move across the membrane. This was our idea of transport, but now we say that from matrix to the outer membrane, when they are changing compartments, say for example, as in the diagram, then this is called as vectorial transport. By this we mean that it has two parameters, magnitude and direction. That is, the number of protons which have to be transported would be fixed, and which side, which means from the matrix to the outside into the cytosol into the outer chamber, or the opposite one. So, that means this is a vectorial transport. Many workers would like to call therefore this chain as electron transfer chain, because the electrons are actually being transferred from one electron carrier to the other. We should thank ourselves that ETC is a common abbreviation for both. So, there is not much turmoil in literature of changing the terminology. ETC is the same. We should also understand by oxidation reduction that how are these electrons and protons actually moving. For this we have to have a knowledge of the redox potential or the oxidation reduction potential. It is abbreviated as E, and it is a measure of the affinity of a substance for electrons. We will measure it relative to hydrogen. The basic idea we have to remember is with ease, because then understanding of the whole electron transport chain will also be easier for us, that if you have a positive redox potential, that means this particular carrier has a very high affinity for electrons as compared to hydrogen, and it would be a good electron acceptor from hydrogen. And a substance or an atom which has a negative redox potential has a lower affinity for electrons and then does hydrogen, and it would denote electrons to the hydrogen forming or the proton forming hydrogen. So, on that note, NADH that we are talking about is a very, very strong reducing agent. It has a negative redox potential and it has a tendency to donate electrons. On the other hand, oxygen is a strong oxidizing agent since it has a positive redox potential, and therefore, it has a tendency to accept electrons. It is very logical to assume therefore that the electrons will always pass from a region of a higher negative redox potential to a lower negative redox potential. In other words, from a lesser positive redox potential to a more positive redox potential. I only wish to tell you in this slide as to what is the relationship between the redox potential and delta G, which is the free energy or Gibbs free energy. Why do we take this concept into account? Because of the fact that ultimately the electron transport chain has to provide so much of energy through the passing of electrons, so that they can trap this energy in the form of ATP through which life survives. So, we find that the Nernst equation tells us that the energetics of the oxidation of NADH, when it gives rise to NAD, that is NAD plus, the delta G is minus 52.6 kilocalories. Here I should also mention that if we have a positive delta E, that means a positive redox potential, it means that the delta G is negative. And when the delta G is negative, then it is characteristic of an exergonic reaction. If you remember, we discussed at length this concept in the very first episode when we were talking about the introduction to respiration. And if we were to compare this figure of minus 52.6 kilocalories to a mole to the actual energy which is needed for the formation of ATP, which is just 7.3. You remember, we had even modified this figure to 9.5 according to the recent findings, and we also then reached a conclusion that 52 minus 70 as compared to plus 7.3 or 9.5 is a huge difference. So, that means the amount of energy which is available is sufficient enough to make many ATP molecules. But then The whole process has to be in steps. It cannot be a one step process, because if it is so, then the whole energy would be uncontrolled and it would not be harnessed, it would not be channelized in the form of ATP. It could perhaps be produced as heat. We will talk about those interesting aspects also. The whole story therefore revolves around two facts. The standard redox potential of NADH is minus 0.3 volts, which means a highly negative redox potential, a very strong reducing agent and a good electron donor. 2, the standard energy change for water or oxygen, which is plus 0.8. So, that means the difference according to the Nernst equation here would be to the tune of plus 1.1 volts. Now, this is not a mean difference. A change in so much of redox potential would have a lot of energy in itself. If this entire energy were to be released in one step, it would be problematic controlling it. And the free energy which is now released is sufficient enough as per calculations to produce 4 molecules of ATP. We will ask ourselves a question. We have already actually asked this question that why does it always give 3 ATP, or does it always give 3 ATP? Now, we are talking about 4. This is only a theoretical yield. Actually, we would see a little later that the figures are entirely different. So, that means, to summarize, the electrons have to pass through stepwise downward movement, so that from an high energy to a lower energy level, when they reach the redox potential of oxygen, then only water would be formed. So, this is the answer to the second question that we asked ourselves earlier as to what is the need of an electron transport chain in the first place. Why can't protons and electrons directly combine with half a molecule of oxygen to give rise to water and be through with it? It is not so simple. It has to pass through an electron transport chain. The second aspect which we should know is what are the carriers for electrons, because electrons cannot move by themselves. They have to be carried by certain carriers. So, metals are the electron carriers, and out of which iron is the most important form, and it exists in the electron transport chain as heme or as an iron sulfur complex, and it is also present in the ferrous and the ferric forms. So, it can easily show the oxidation reduction reactions. For example, if the ferric form of iron combines with 2 electrons, it would give rise to a ferrous. We would see a little later, when we look at the electron transport chain a little more closely, we would find that there is one center, binuclear center, which contains copper also, and it is the enzyme called as Cytochrome Oxidase, which has iron as well as copper. Now, in case of copper, copper would exist in 2 forms here, copper B and copper A, and it also would show now the redox reaction of cuprous, getting the electrons and turning the cupric forms. Cupric would take the electrons and it would give rise to cuprous forms. There are, however, certain acids and bases which are good carriers for protons. Protons can move on their own also, they can pass through porins. There are certain proteins which are carriers, we will talk about it very shortly. And on the other hand, our focus of attention now is the carriers which can carry both electrons as well as protons, and they are NADH, because here we would find that a pair of electrons and a proton are simultaneously being transported. And then the quinones, the QH2. But then the difference is that these particular quinones would transfer the electrons in 2 steps. One electron and then it will change into a radical and the ubiquinone would be formed. And a second electron would come later and it would take along with it 2 protons and it would form a ubiquinol. So, if you remember your chemistry lessons, it is a good example of quinone oxidation, quinone oxidation. And then there are certain flavin cofactors which we have, you remember in succinic dehydrogenase step we had FAD. So, the flavin adenine dinucleotide. They also can, they are responsible for the transport of 1 or 2 electrons. Here I should mention that the redox potential of NADH is the most negative as compared to quinones and FADH2. These complexes are membrane bounded, and when we look at the electron transport chain, there are 5 complexes which are bound to the inner mitochondrial membrane. This entire episode that we are talking about is taking place at the inner mitochondrial membrane. The outer mitochondrial membrane was permeable. So, anything selective which is going to happen in the internal membrane or the inner mitochondrial membrane. These different complexes can be viewed in the form of islands in a membrane sea, which is the classical sentence that was used by Singer and Nicholson when they gave the fluid mosaic model of a plasma membrane, and they communicate with each other indirectly. That means there is no direct relationship. The first complex has NADH and quinone reductase. The second complex has the succinate Q reductase, the enzyme complex, and then you have the quinone cytochrome C reductase complex, followed by the fourth or the last complex on the membrane called as cytochrome C oxidase. We will spend some time on it after the discussion. And ATP synthesizing sites, where ultimately ATP is going to be formed via certain proton circuits or proton gradients, that constitutes the complex 5. What we have to appreciate is that the electrons are going to move from a negative redox potential towards a lesser negative redox potential, which means that is the standard potential is 0.3 minus to 0.8 plus to form water. This is the ultimate journey. If we were to appreciate the components of electron transport chain on a membrane in a topological form, these are the different complexes that we are talking about, and these particular complexes starting from 1 from the cytosol, again they have a proton exit into the cytosol, and then the complex 2 which is linked, the complex 3, the complex 2 would come from succinic acid, succinate fumarate. You remember in the Krebs cycle we said that when succinate changes to fumarate, FADH2 is formed and it yields only 2 ATP molecules. Now is the time to explain how. And then this is the 5th complex where the ATP is generated. The 4th complex is also very, very interesting because it has the binucleate center which contains iron as well as copper. The summary therefore is that if you are talking about the redox potential, our journey will begin from 0.32 of minus electron volts to 0.82 plus, where oxygen is going to be giving rise to water. And this journey is seen in the form of islands of different types of the carriers of electrons, starting from the first complex to the 4th, and ultimately the 5th complex which has F0 and F1 particles, they are the oxysomes or the respiratory assemblies, where ATP synthase is present, and this very complicated enzyme complex ultimately gives rise to ATP, and this is what is called as oxidative phosphorylation. If we were to look at the scheme of the movement, what really happens is any reaction we take, NADH is formed, say for example in glycolysis or in Krebs cycle. Now, this is taken up in the complex 1, it is now oxidized back to NAD plus protons, and it donates its 2 electrons to FMN. FMN is flavin mononucleotide, which is a non-heme iron, and it has the hydrogen ion complexes. And there is a further downhill transport to coenzyme Q. If you see carefully, the yellow markings are of the succinate coenzyme Q complex, which is 2, which is not in the straight line or the scheme of things. It comes from the other side, and then the first is connected directly to the complex 3 and complex 4. Looking at this simplistically, let me just tell you how the whole thing happens, the whole process. Any substance which is now reduced, say for example, any of the compounds which has been formed in Krebs cycle, it would give rise to the other compound by a cyclic transfer. The cyclic transfer of electrons will take place from section 1 to the complex 2, and between NAD and FMN, one molecule of ATP is formed. We have to see in details how is it formed. That means there must be some energy difference between the two in the redox potentials, and then comes the cytochromes after coenzyme. The succinic acid or FADH2 comes somewhere here. So, that means this is the first site of phosphorylation, this is the second site of phosphorylation where the cytochromes are there. The sequence of cytochromes is cytochrome B, cytochrome C, cytochrome B, B1C, A, A3. Coming to A3, we have a bi-copper complex, and this bi-copper complex now has copper as well as iron. And finally, the third phosphorylating site is there at the end of complex 4, and half a molecule of oxygen, 2 electrons plus 2 electrons from the electron transport chain, and 2 protons finally give rise to water, and the whole chain reaches 0.8 electron volts of plus, which is sufficient to form water. So, that means this is the answer to the question why oxygen and the electrons and protons cannot simply react and give rise to water. So, that means they have to pass through a descending series of the descending redox potentials so as to give rise to water. And we summarize therefore that the oxygen plus 4 electrons would give rise to 2 oxygen radicals, which taking 4 protons would give rise to 2 molecules of water. Two very important questions emerge from this discussion so far. What is the final acceptor of electrons in the electron transport chain? Oxygen. And what is likewise the final acceptor of protons in the electron transport chain? It is again oxygen. So, oxygen is the final acceptor. You remember, throughout the process of aerobic respiration, we have nowhere mentioned the role of oxygen. That when we had a shift from anaerobic to aerobic respiration from glycolysis and pyruvic acid, the whole process of oxygen involvement has come at the ultimate stage. So, this is the process of electron transport chain. Let us spend some time on the very important cytochrome oxidase complex, and there are many things that one need to remember. Cytochrome C oxidase, which is at the complex 4, has 2 cytochromes, cytochrome A and copper containing copper A and copper B, A and A3. The cytochrome oxidase complex is the most sensitive to poisons, because it has an allosteric site. So, it is also sensitive to carbon monoxide, azide as well as the cyanides. And if we see in the equation, the ferric and ferrous conversions are there, and simultaneously the cuprous and cupric conversions are also present. There are so many inhibitors in the chart that we can see. For example, rotenone and amytal, they are going to inhibit NADH Q reductase. That means they are going to act at step number 1 and they are going to stop respiration. That means the transfer of electrons from NADH to FMN, and then the whole thing would stop. But then if we are coming through the complex 2, the complex 2 downwards process will still continue. So, that means we still have a channel open. Actimycin C and actimycin A also act on Q, that is the cytochrome C oxidoreductases. We should spend some time on the cytochrome oxidase, because cyanides, azides and carbon monoxide, they are very, very potent poisons. We also know by various tales and by scientific facts also that cyanides are one of the most potent poisons for respiration. Actually, cyanides are acting at the competitive inhibitory level of the enzyme cytochrome oxidase, and they are going to block the enzyme, thereby rendering it absolutely ineffective. The water formation stops, the electron transport chain stops, and so does the ATP production, and it finally leads to seizure of respiration and of life. All of us remember the Bhopal Gas Tragedy, which occurred on the night of December 2nd and 3rd, 1984, when the Union Carbide factory released huge amounts of methyl isocyanate. Now, this is a situation where the isocyanate acted with the cytochrome C oxidase, and it is recorded to be the worst industrial disasters in human history, taking a toll of about 3,800 lives, and many thousands are still paralyzed and having lot of problems. What is the relevance of the discussion here? Have we ever thought? What happened to the plants and the bacteria of Bhopal? Did the entire vegetation also get wiped off? Perhaps no. That means there is something like a cyanide resistant respiration also. Now, that is a very interesting aspect, because there are certain plants, many plants for example, have a special type of NADH reductase, which is not sensitive to rotenone. If it is not sensitive to rotenone, and also we have they also have a cytochrome oxidase complex, which is not sensitive to cyanides. So, that means this pathway would be a little divergent from coenzyme Q, and it would give rise to products, perhaps less of ATP, but then the cyanide problem is circumvented. So, many bacteria, many archaebacteria, many plants, all of them, they are showing cyanide resistance. Let us not forget, life originated in an atmosphere of highly reducing agents and poisonous substances like cyanides and ammonia, and today they are the biggest poisons. That means this type of respiration must have originated in evolution much earlier than the present cyanide sensitive respiration. Another very important aspect is that these particular plants, if they are not having the normal electron transport chain and a shortened electron transport chain, then all the required steps are not there, and therefore, as I said earlier, what is going to be the fate of the energy? It is now going to be released as heat. So, such plants like skunks, like aroids, and also potato, they are cyanide resistant, so are certain bacteria, and they release heat. We know of a skunk cabbage which is growing in snow and which can increase the temperature from 10 to 15 degrees, and 5 degrees temperature is quite a bit, so that even snow in which they are growing gets melted. And when the temperature of the body of a plant increases, we would find that the odoriferous alkaloids in a plant, they would be released as volatiles. So, these are the plants which stink, which give a very foul odor, and we know that in nature it is not only the sweet fragrance which attracts, it is the foul odor also which attracts the pollination agents like sawflies, like beetles, and like various insects. Needless to say, such type of pollination will take place even in dark, because it's only a question of smell, and it's not any attraction of color, so that sunlight is not required. We should never forget the example of Rafflesia. Rafflesia is the largest flower that we know, and you cannot stand by it because it is so very foul smelling, and it also releases this odor on account of cyanide resistant respiration, and it attracts sawflies at night for its pollination as visitors. Another very important aspect regarding the uncouplers of respiration is that there is one compound called as 2,6-dinitrophenol indophenol, which is also abbreviated as DNP. This compound, as we see, acts somewhere near actin-myosin C, between cytochrome B and cytochrome C, and it uncouples the proton movement in such a way that the electron transport chain takes place, but ATP is not produced. And when ATP is not produced, then what is going to happen to the huge amount of energy? It is released in the form of heat. There is a protein even in our body called as digenin, and this particular digenin or thermogenin, as we call it, this thermogenin is responsible for increase in temperature. The protons are directly taken up by the thermogenin, so that there is no proton pumping, there is no proton gradient formed, and the amount of heat which is released is dissipated to the environment. This happens in nature also. You know, a newly born human baby or a newly born animal can sustain itself because a portion of its brown fat is going to show this type of respiration, that is no ATP production but heat. This is called as non-shivering thermogenesis. This is how some of the vital and sensitive organs and tissues of a newly born are protected. Do you remember? How does a polar bear remain intact in such a huge amount of snow and such a low temperature? It has to hibernate, and when it does, then a part of its brown fat also shows what is called as non-shivering thermogenesis. So, this is again a corollary of the uncoupling agents of the electron transport chain, and they are now engaged in production of heat to sustain or to protect themselves rather than to generate ATP for various metabolic activities, because they are really not required, and ATP is not required when an animal is busy protecting itself from the outside environment. Coming to the aspect of oxidative phosphorylation, first let us try to appreciate the structure of the fine structure of a mitochondria. A membraned organelle, a semi-genetically autonomous organelle, has the outer membrane which has porins. We have discussed in the last episode as to how there are various transporters and they are going inside, and then there are these racket shaped bodies which are present on the inside. The two faces are there. The face which is towards the matrix is called as the M phase, and the phase which is towards the intermembrane space is called as the C phase. These two faces of the internal membrane have different permeability properties, and the racket like assemblies which are there, which have a rotor at the knobs inside, they are called as the F1 particles, the F0 particles. The word F comes after their discoverer H. Fernandez Moran, who employed very sophisticated techniques of freeze fracture and silver staining to find out that these particles actually exist, and they are the respiratory assemblies. These are the assemblies where ATP is generated. Oxidative phosphorylation and ATP synthesis would take into account the basic idea that the energy which has been generated through the electron transport chain has to now get utilized in the formation of ATP. So, that means some sort of pumps are operative at this place. What really happens is that as a result of the electron transport chain, the protons are pumped into the intermembrane space. When they are pumped into the intermembrane space, this is going to result in a huge accumulation of protons in the intermembrane space. As a result, there is a large proton gradient which is going to be formed, and these protons would like to come back into the matrix. They cannot. This is what I said when I mentioned to you the differential permeability properties of the inner membrane. The M phase would allow the outward flow of the protons, but the C phase, which is towards the cytosol, would not allow the protons to enter. The F1 particles are located in the inner membrane. There are specialized channels which are there. So, the protons can move only through these channels along a very high proton gradient, and as a result the coupling or the osmoticum is going to generate a proton motive force. So, that means the H ions are first pumped out into the matrix by 3 pumps. The M phase is permeable to protons, fine, it goes outside. Pumping of the protons generates a very high concentration. The electrical potential now becomes positive and a huge amount of protons are accumulated, but the C phase is impermeable. So, they will like to go only from a place where they are allowed to grow, and that means the generation of an electrochemical proton gradient is formed only through the proton channels which are present on the F0 particles, and this generates a proton motive force. And when this generates a proton motive force, then ultimately when the protons reach the F1 particles, then they activate the enzyme complex ATP synthase to generate ATP. So, in other words, the process of ATP synthesis is chemical, no doubt, but then it cannot occur on its own. This whole process is driven by an electrochemical osmotic gradient, and this is the basic idea what Peter Michel proposed when he said that this whole formation of ATP is chemiosmotic coupling hypothesis that he gave. In 1961, his work was almost condemned and laughed at, because this was a novel way of having a chemical reaction, that is by an osmotic means, but then later it turned out to be true, and this is one of the most important discoveries in the whole of the chapter of respiration, where, as we can see in the diagram, there is a sealed compartment of the matrix, and how the protons are going out into the outer membrane space, getting themselves accumulated, creating a proton gradient, and ultimately going through the proton channels and generating ATP. So, this is the basic idea of chemiosmotic coupling. An overview of the ATP synthesis tells as to what are the different areas of ATP synthesis, the different complexes 1, 2 and 3, and the 4 and the 5th one we have just discussed. This is the F0 and F1 particles. Coming to the last question as to what is this whole business of 36 and 38 ATP. You remember the two molecules of ATP were formed directly in glycolysis. There are two molecules of NADH which were formed in glycolysis. We said, okay, we will see when the electron transport chain comes, and now these particular NADH which are cytosolic also need to be reoxidized. We have already discussed how does the reoxidation take place, but then what is going to happen to the cytosolic NADH molecules? They can freely enter the outer membrane, because the moment they come in contact with the C phase of the internal membrane, they cannot enter. If they cannot, how would they enter the electron transport chain? So, that means there are two mechanisms of reoxidation of cytosolic NADH which are called as the shuttle mechanisms. There are two types of shuttle mechanisms, namely glycerol phosphate shuttle and the malate aspartate shuttle. Looking at the first shuttle, we find that this involves FADH2, because the NADH cannot enter. So, when the NADH enters inside, it cannot, and what will happen now? The reaction will now turn into dihydroxyacetone phosphate. It will give rise to glycerol 3 phosphate. Now, this NADH we are talking about, it cannot go alone. So, if this NADH has to go inside, it has to first convert into NAD. The glycerol phosphate will enter. That means dihydroxyacetone phosphate cannot enter. It has taken its NAD from somewhere else. This NADH gets oxidized, glycerol phosphate goes inside. The moment it goes inside, it cannot enter the inner membrane. FADH2 will take H from glycerol phosphate, convert itself into FADH2, and the dihydroxyacetone phosphate will come back. So, this whole cycle goes on and FADH2 is formed. We have just now seen in the electron transport chain that when this FADH2 enters the electron transport chain, it would give only 2 ATP. So, that means 2 ATP are formed, and the net gain of ATP would be 36. If you remember, 2 FADH2 will give 4 ATP and 2 direct are there. This is the whole process for glycolysis, and 24 ATP have already been formed through the ETC. So, that means it would give rise to the total number of ATP molecules 36. Unfortunately, this type of shuttle operates in our brain and in the nervous tissue. So, this is the story of 36 ATP, whereas in our heart and liver and also predominantly in plants, there is another very intelligent mechanism which can even give 38 ATP. What really happens is, it takes into account the involvement of malic acid. Malic acid can enter the cytosol through the inner membrane, because its molecular weight is less than 10,000 Daltons. So, once it enters the malic acid through a transporter, the alpha ketoglutarate transporter, and it will now undergo a reaction, malic acid giving rise to oxaloacetate. and NADH is formed. So, that means it is something like writing a check outside and not entering a bank, or taking a blank check inside the bank and then filling it up. So, malate aspartate shuttle does the latter, and finally it gets converted into aspartate. Aspartate comes back and is reconverted into oxaloacetate. So, this NADH which is formed inside the cytosol would again give rise to 3 ATP. So, that means the net gain of ATP here is 38. All the other calculations we have done before, it was a question of whether the cytosolic NADH would give rise to 2 ATP or would it give rise to 3 ATP. That makes the difference. However, the more interesting aspect is the one which has recently been given by the scientist Paul Boyer. He says that the ATP is synthesized by the F0, F1 complexes which are in the form of a rotary engine. They have even described the different types of proteins which are present in the F1 particles and in the F2 particles, and they are making a 3-dimensional model in the form of a stator and a rotor, and it is all because of the static components and the mobile components of this rotary machine that we have a proton gradient being generated, which finally gives rise to ATP. Coming back to the final summary, our conventional view we have just described of 36 and 38 ATP molecules. However, very recently the stoichiometry of ATP synthesis has been reinvestigated with more sophisticated equipment. As we know, nothing is static in science. Anything which we find correct may be subject to modification. So, now it has been found that one molecule of NADH would give rise to 2.5 ATP and not 3. And likewise, one molecule of FADH2 would give rise to 1.5 ATP. So, our calculations have all gone haywire. We were all the time fighting between 36 and 38. Now, the matter is altogether different. As per new calculations, we are now wherever we had 3 as far as 1 NADH was concerned, we have 2.5. Wherever we had 2 for FADH2, we have 1.5. So, now if we are talking in terms of glycerol phosphate shuttle, the net gain of ATP would be 30, and the net gain of ATP in malate aspartate shuttle would be 32. So, there are so many questions which have emerged out of this discussion as to the calculations of ATP have been reinvestigated, and now the story would be between 30 and 32. There are other forms of respiration also which we should remember, because if you were to ask a question that our entire story revolves around NAD, what would happen if the products of photosynthesis like NADP come? Then we have other mechanisms called as pentose phosphate pathway, which are there in the older tissues, and the Entner Dodorov pathway, which is existing in the bacteria. So, I have just tried to summarize what the recent findings regarding the mechanism of ATP generation are. One should remember the watchword for the entire discussion is that the production of ATP is a chemical process, all right, but then it is driven by an osmoticum. So, basically it is a chemiosmotic coupling. Thank you.
With this note, thank you sir, thank you so very much for explaining once again in detail about the electron transport and ATP synthesis. And so far now we have 3 lectures on cell respiration. If you wish to access these particular lectures that is on cell respiration, you can definitely access these lectures with the help of the YouTube, as we always say that we used to upload our lectures daily on the YouTube. Now, very soon we are going to start a very new series that is on the genetics. So, because we have very less time left over here, sir, what genetics are and what is the study about?
Well, I always start my lecture on genetics by saying that we are like our parents yet different. So that means the characters which we take from our parents is transmission of characters, which is heredity. And we are not photocopies of our parents. So that means there is some variation. So heredity plus variation would become genetics. I would like to stress upon the classical aspects of genetics, which are perhaps long forgotten, and then gradually come to the modern or the molecular genetics.
Definitely, we are going to conduct series of lectures on classical genetics and genetics as a whole. We would like to cover for you, so that you would be able to study all the material sometimes which you sometimes tend to skip in your classes. But I hope that these lectures would be beneficial for you in studying as well as enlightening yourself. With this note, thank you, sir. Thank you so very much for being the part of the EduSet lecture.